So let's say we have DNA molecule, double helix structure, sugar phosphate backbone, the purple part, and then complementary base pairing of the nitrogenous bases, adenine, thymine, guanine, cytosine, right? A segment portion of the DNA is called a gene. Then DNA, when it's wound up, wrapped in proteins, highly coiled, prior to cell division, it looks like this chromosome. But actually, normally, it exists as chromatin. This part, okay. This is another one. Not sure whether this helps. Maybe this one. Okay, with that, we are ready to start today's molecular genetics. Okay, so in your textbook, we actually showed this DNA molecule wrapped in proteins, and then it is packed as chromatin in the nucleus. Prior to cell division, it might appear like the chromosome. So what is a DNA? Deoxyribonucleic acid, right? So that's where all the different segments of DNA are, all the genes are stored. But this DNA molecule consists of polynucleotide. What do I mean? This one strand, one whole strand, two strands of polynucleotide. Polynucleotide, many nucleotide. What's one nucleotide? One nucleotide consists of the sugar ribose, deoxyribose sugar, one phosphate group, and a nitrogenous base, which can be either A, T, G, or C. Normally in a DNA. Okay, the copied one will not have T. The copied mRNA messenger RNA will have U. So give an example of one side of the DNA. Okay, so be careful how it's drawn up. So it is attached to the first carbon, fifth carbon, 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. First and fifth carbon. Because sugar at the end of the day is, for example, glucose is C6H12O6. A ribose sugar happens to be a 5-carbon sugar. Okay, this one is talking about the complementary anti-parallel. You see the phosphate is 5' here, 3' here, 5' here, 3' here. Okay, so common question, complementary base, maybe they give you the top strand, then you match the bottom A with T, G with C, that kind of question. So they may give you the top and bottom, and always remember A and T, same ratio. So if I give you like, oh, adenine is 30%, then you know that A thymine also 30%. 30 and 30, 60, left 40. So divide by 2, 20, 20%, that kind of thing. Okay, let's talk about genes. Here, see, definition, gene is a sequence of nucleotides. It controls the production of polypeptide. So same thing, I can have a gene that is for insulin. So this insulin gene will contain a specific sequence of nucleotides that's going to control the production of the insulin polypeptide. So if I'm very specific, then maybe I need a gene to produce the 2 polypeptide to make the insulin protein. Okay, don't need to know the details, but the idea that every 3 bases, every 3 nucleotide codes for 1 amino acid. 3 nucleotide is also called 1 codon. Okay, and since all the bases will have all the bases, the sequence, that is also the genetic code in the DNA. Okay, so the codon, every 3 nucleotide is 1 codon. 1 codon codes for 1 amino acid. So the sequence of the genes, and therefore the sequence of bases, will determine the sequence of the polypeptide form. So how are we going to make protein? So don't need all the details, but the idea that, firstly, you must copy the DNA. That is called transcription. Secondly, whatever that you copy is called the mRNA, messenger RNA. So after you copy this, at the ribosome, you need to translate to the actual polypeptide. So the main terms to know is transcription, translation. When from the DNA, you undergo transcription, you make the mRNA, then you undergo translation to make the polypeptide. So I'll give you an example. From the insulin gene, so the DNA contains maybe a segment that is the insulin gene. Transcription will produce the mRNA. Translation will produce one of the polypeptide for insulin. That's all.